Okay, good afternoon. May we start the sessions? Good afternoon, Prof. Alex. How are you?
Very good, thank you. Good morning.
Great.
Well, here is
Good morning.
morning
Yes.
or afternoon for you.
Yes, good morning then.
Yeah, it's a pleasure to be here again. Yeah.
Yeah, it's really nice to have you in this session, Prof. Alex. So, is it okay for me to address you as Pak Alex? Because in Indonesia, we usually call Pak.
Yes, sure. Yeah.
Okay, Pak Alex, it's nice to have you. So, let's start the sessions. Can we start the session possibly?
Yeah, sure. Please.
Okay. So Assalamualaikum warahmatullahi wabarakatuh. Good morning, Prof. Alex, and also good afternoon, Bapak Ibu, ladies and gentlemen, all the invitation and participants. It's really nice to have you today. And then welcome to 3-in-1 guest lectures in Microbiome in Extreme Environments held on Monday, 4th of September 2023. And my name is Dian Islami, and I will be the master of ceremony for today's sessions. And then for your information, that actually the 3-in-1 guest lecture is the sequence of the 3-in-1 programs that is the initiations of the international engagements and collaborations with international lecturers, scientists, and national practitioners to embrace and engage wider knowledge and also to raise up positive views toward the issue happening globally. And then in this opportunity, it is a really an amazing chance for us to collaborate with King Abdullah University of Science and Technology. The honorable mentions the head of Department of Plant, Pest, and Disease, Dr. Lukman Kurota Aini. The guest lecturers for today, Prof. Alexandre Soares Rosado from King Abdullah University of Science and Technology, all the lecturers, staff, and students who joined today's sessions. And then in these occasions, I would like to also inform you that we have several agendas to follow. The first one is we will like to listen together to the opening speech by the head of Department of the Plant, Pest, and Disease, Dr. Lukman Kurota Aini. And then we go to the main session of the guest lecture, and this session will be led by our moderator today. We have Dr. Sibli here as the moderator today, and then we also have a discussion session. So in these sessions, we would like to encourage all the participants here to make some questions and ask questions to Pak Alex as our guest lecturer today. And then after the discussion session, we will have the closing sessions. And then to take the advantage of times, let's start the first agenda. So we will listen together to the speech by the head of Department of Plant, Pest, and Disease, Dr. Lukman. Dr. Lukman is here.
Okay.
Yes, okay. Pak Lukman, time is yours, Bapak.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon to all of the audience, and good morning to Professor Rosado. Nice to meet you again.
Yeah. The pleasure.
Okay. So Alhamdulillah, so we can meet in this occasion, a special occasion, because this afternoon we have a 3-in-1 program. This is one of 3-in-1 program that invite highly qualified and professor from academic from abroad. That is Professor Rosado, this distinguished professor, and so we will join this very interesting topic also. This is about the microbiome in extreme environment. So I think this is very interesting and important to us, particularly how to get the benefit from the microbiome in environment, and particularly in an extreme environment. So actually, we in our department also, we have a lot students that do the exploration of microbes, fungi, or also bacteria, that we try to find the benefit for particularly for the plant. That's why I think this topic is very interesting, and I hope this can improve the knowledge and also the skill of our student, particularly for the master student, that um, in the plant pathology and also entomology study program, and also the our other graduate program also follow this lecture. So at this time, I would like to thank to Professor Rosado who have give uh, a lecture today, and also to the participant and also Budian and possibly who arranged this lecture. I hope all of the audience can get an insight how to improve the research related to the microbe. I think that's all that from me. Maybe we also the next lecture. We meet in the next lecture. I think that that's all. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Pak Lukman, for the opening speech. Now, before we go to the main sessions, I would like to invite for documentations, maybe to make it more officials for us before we start the lecture. So let us take picture together with Alex. And then maybe I need to help from the committee to take the picture. So I also would like to invite all the participants here to turn on the camera so I can see you. Okay, are we ready to start the documentation or take picture? Okay, so I would like to count 1, 2, 3. Maybe one more. 1, 2, 3. Okay, thank you so much, everyone. Yes, so because we have already take pictures, it means that the lectures will be start because it is officials. Okay, so after the opening speech by the head of department, so we would like to start directly about the main sessions. As what I mentioned before, that this main session will be led by our moderator today. So I would like to invite possibly as the moderator for today. But before, I would like also to read the CV of our moderator today. So please share the CV. I'm sorry. Okay, so our honorable moderator for today is Dr. Muhammad Akid Sibli. He had the bachelor degree in Universitas Brawijaya on 2010, and then for the master degree also in Universitas Brawijaya on 2019, and then for the PhD or doctoral degree, he had the doctoral degree from Hokkaido University in 2020, and his expertise is in the biological control, metagenomic, and arbuscul mycorrhiza. And then there are some selected publications. The recent one is in 2022, the screening of the yeast isolate for the biocontrol of Sclerotium rofsing, Biodiversitas Journal Biology of Diversity. And then community services is also the recent one about the extensions in the pengelolaan penyakit akar gada, upaya pengurangan penggunaan pestisida dan pada tanaman sawi books, menuju good agricultural practices pada kelompok taruna tani sumber berantas batu di tahun 2023, in the year of 2023. So that I would like to hand over these sessions to our moderator today. So possibly time is yours.
Thank you so much, Dian, and everyone. Hello, Alex. How are you? Long time no see.
Very good. Good to see you. Yeah, good to see you again.
Yeah, good to see you too. Next year, probably we can meet in person. We can visit our university. We're very happy to have you here, and we can start some initiation, research, education, and something related to research, especially in microbiome. So everyone, I'm Muhammad Shibli for moderator today's webinar. Warm welcome to all of you. So we are here today to listen to and engage with Professor Alex from King Abdullah University of Science and Technology to share ideas on how microbes lives in the extreme environment. Maybe from the committee, you can share the CV of our lecturers so I can read some information about Professor Alex. So Prof. Alex had his master program from Brazil, Camafilo University, 1989, and continue his PhD in the Wageningen University for Plant Research International in the Netherlands, 1997. And he had interested in centered around the study of microbial diversity, evolution, new microbial metabolisms using conventional tools associated to multi-omics approaches to understand the evolutionary history. So we will listen about the new concept about microbe community. And I think it will become interesting for all of you to understand how this complex system works in the field, especially from extreme environment. And this is the selected publication from Prof. Alex from 2020. We had extreme environment, a source of biosurfactant for biotechnological application, extreme fields, journals. So this is our opportunity to listen more and learn about this topic. Thank you for the committee. So Alex, we will have a 45 minutes lecture from you, and then we can continue with the discussion with the audience to talk further about their understanding on these topics. Can we start?
Yes, sure. Very good. Thank you so much for this amazing opportunity to be here again with you guys. It's my second year now participating in those 3-in-1 series of seminars. And last year was super impressive and amazing. I had a very good moment sharing the microbiome field and some of my experience with microbes with you and your students. So it was really amazing. So thank you again. Thank you, Mohammed Sibyl, and also for the department and the committee for this invitation and the opportunity to be here again. I can start sharing my screen. Are you three? Let's see.
Yeah.
Yes, I think it is one. Can you see or no?
Yes, yes.
The presentation, yeah? Okay. So I will open the... Okay, perfect. Thank you. Thank you very much. Yes, so it's my real pleasure to be here again. And today I want to share a little bit of my experience with those very interesting microbes that not only survive, but that they thrive, and they can thrive in what we consider a very extreme environments. And we can learn a lot about our life evolution on Earth, on our planet, and we can also explore, their huge potential of extremozymes or new enzymes to be used in several different applications in biotechnology. So they are super interesting. There is also a very huge interest nowadays from, everybody is, I think, following the huge achievements in the boost of different space agencies like NASA in India, now a very successful launch and landing in the south pole of the moon. Emirates, Saudi Arabia now has 2 astronauts that went to the International Space Station. So there is a huge and amazing race to understand a little bit more about space and space research. the microbes, and especially the microbes from the extreme environments, they can play a very important role in all these, possible space colonization, not only exploration, but colonization. So, yes, I'm a professor here at KAUST. I joined KAUST almost 3 years ago. I'm from Brazil, as Silvio was explaining, showing my CV. I was full professor in Federal University of Rio de Janeiro. I spent some time in the Netherlands doing my PhD in Wageningen, and after that, in Wageningen was my first contact with the Indonesian food. It was amazing. I had a lot of colleagues from Indonesia there, and I used to have the real amazing Indonesian food. Not only with my colleagues from Indonesia that were living in Netherlands at that time.
Next year, Alex. Next year.
Yeah,
but I will prepare for you.
Okay, yeah, I'd be happy with that. And after that, I moved to California, and I was there for about 4 years at UC Davis, University of California, Davis. And before joining KAUST, actually during the pandemic in 2020, end of 2020. So it was, we all went to those crazy times, and but it was amazing to be here. And so I'm really happy to be working here in Saudi Arabia, and especially in KAUST, because it's a very special place. This picture actually is from my, literally from my office. We have amazing sunsets here in the Red Sea. Can be a little bit hot during the summer, but it's not big deal for someone that came from Rio de Janeiro in Brazil, so it's okay. But after the summer, the weather and the temperature is amazing. So it's very good. I have my lab here, and my laboratory here at KAUST is dedicated to basic research, but also we have a very strong arm, a very strong line of research dedicated to application, the biotech application of microbes. And actually, in my group, we've been working on several different ecosystems, not only extreme environments, but also human microbiome. We have some very interesting work and studies, previous working on human microbiome, and now we have a lot of new students working on a human microbiome. We have several different other ecosystems, and not only extreme environments, but the extreme environments, they actually, I don't say that I have that I prefer to work with extreme environments, because I have several other different research lines and students, but actually the microbes that you can isolate, or the metabolisms that you can find in such very extreme environments, they are very special, because they can be unique. And so this actually catch my attention, and I have a bit of huge passion for those specific microbes, or particular microbes. They are very interesting. And in my group, we like to work at ecosystem level. So we try to use all the omics that we know nowadays, metabolomics, transcriptomics, the metagenomics, metatranscriptomics, metaproteomics, and also metagenomics, to try to understand ecosystems and the biomes, and not only at taxonomical level, but also the functional diversity and the potential for those microbes, to perform different biogeochemistry cycles, and how they interact with hosts and so on. We also have a very strong microbiologist, and actually old school microbiologist. So I love to isolate microbes. And so we have a very important research line based on isolation and cultivation of those microbes, especially mainly bacteria, and also fungi. And so we also perform studies at organismal level on those microbes. And of course, I do love to see not only the fundamental, not only the basic research that's super special, and I love that, but I do love to see those microbes that we've been working for so long, and we are still studying those microbes, how they can perform, how they can be applied in the real world to perform some biotech applications. So we also have a very strong research line, and I have some students and research scientists working in a more dedicated fashion to biotech solutions. So this is the application of our work. And we've been talking about microbes and the microbiome. And then what is microbiome? And there is no simple definition of what is microbiome. We have several different definitions, and different groups usually they will stick to one particular definition. But I like to follow the guidelines proposed by Gabriel Berg and collaborators in 2020, because I think it's more complete. So they wrote a perspective paper together, and they published this in the Microbiome Journal. And so it was Gabrielle Berg from Austria. She was leading the publication, but with more than 10 to 15 different microbiome scientists from the completely different fields. So some scientists working on human microbiome, on some others in soil microbiome, some other experts in plant microbiome, and environmental microbiome, and also insect microbiome. So different expertise, they sit together and they try to extract a definition, a consensus definition. This is, of course, is super difficult, super tough, but they came up with a very interesting definition that the microbiome we can define as the everything. So it's not only the microbiota. Microbiota is a compartmentalized section. So we have the bacteria population or community, bacterial community. So this is the microbiota of bacteria. They are part of the microbiota. We have the archaea. So this is another compartment, and that's another microbiota. We have the fungi. So this is another compartment. So we have the microbiota. We have also, we can include the viruses. I mean, because they can behave as living beings when they have a host to replicate their material, genetic material. And when you put everything together, all the different parts of the puzzle, so all the different compartments, bacteria, fungi, archaea, viruses, you can also include some other microeukaryotes. Some people also include in the protists in those groups, algae, microalgae, something like that. And when we combine everything, and we also include the interactions between them and among those different groups, and also the interactions with those different compartments in the ecosystem, the biome, that can be a soil, or a plant, or a gut from a human, or mosquito, or so doesn't matter. The ecosystem itself can be a soil or a human, but when you have this particular ecosystem or biome, and you have the microbes associated, interacting and exchanging and communicating, especially through metabolites, we have the microbiome from that particular ecosystem. So that's why we have the soil microbiome. So all the microbes and their interactions within the soil ecosystem. This is why we have the human microbiome. All the microbes and different compartments, so bacteria, fungi, viruses, and so on, inside human, or in the skin, at the skin, or something like that. The same is for plant, the same is for insect microbiome. So it's very diverse, and this is a more broad definition that I think it's quite interesting. And why we are talking a lot about microbes and microbiome nowadays. We've been following out the different publications and in top ranked journals, talking about the microbes and the power of those microbes in how they can help not human health, but also ecosystem health and so on. And some scientists, microbiologists, they like to say that we are living now in what they call the third golden age of microbiology, because we have the beginning till Louis Pasteur, Anthony Leeuwenhoek, and now those pioneers till Pasteur. So we have the first golden age, so the beginning. So the first thing, the microbes and the identification of microbes, and we have the second golden age. This came with especially the molecular tools in the end of 50s and the beginning of 70s and 80s. So the discovery of the DNA structure, the recombination of DNA, all those tools. And now we have what they say, the third golden age of microbiology. And this came together after the advent of the next generation sequencing platforms. So nowadays, it's really impressive, the potential to explore and to detect sequence of DNA or RNA or proteins in different environmental samples. And this, of course, it's, there are some graphs showing the number of publications and patents when we have the advent of the first next generation sequencing platform. In that time was the 454 from Roche, was a long time ago, into actually the first paper using a powerful sequencing machine was published in 2005 using soil samples and water and soil samples the next year. And after that, we have this boom of publications, and we really start to understand more about the microbes and how they can live in completely different environments. And we also have the metabolomics that actually is booming now. It's really literally exploding the field, because we can understand that also it's still not easy, of course, but we can target now some metabolites. And so we can start to understand how microbes communicated to each other and how microbes can communicate with the hosts, through the detection and trying to understand the metabolites they are producing. And so this is, super impressive. And of course, to analyze all those data that we have been generating, we need to rely a lot in bioinformatics. So I would point like the next generation sequencing platforms, plus the increase, the evolution of new pipelines and protocols in bioinformatics. So we have a lot of very impressive tools nowadays that we can really extract, biological information from now those data that we've been generating for a while now. So this is why we are now really talking about microbes and the importance of microbes. And this is one example from a paper from a postdoc from my group and some other students, Junior Schultz, how we can, from an environmental sample, the different approaches and tools that we can use, we can apply nowadays. So we can, from environmental sample, we can go to high throughput sequencing, so we can perform shotgun metagenomics. We can also perform amplicon sequencing, and so we can have information about the taxonomical diversity of such particular environment. And again, can be any environment, can be a human gut, can be an insect, can be a plant or a soil sample or extreme environment, extreme soil, something like that. And you can have taxonomic information. We can also infer the functional potential of that particular microbial community based on the potential, the genes that we can find in that environment, in that ecosystem. And we can also, of course, go directly for RNA, so we can actually detect the genes that are being expressed in that particular ecosystem, in that particular situation. We can also rely on metabolomics nowadays. It's still it's still in the frontier. It's still in the border. I mean, we're still learning how to understand those metabolites, especially particularly when you talk about environmental sample. It's really tough. It's really difficult, but we can see daily advances in the ways that we can really extract meaningful biological information. And we also need to rely in cultivation a lot, because I mean, we can have the sequences and we can extract biological information. You can even apply if you detect some sequences, we don't need to isolate the microbes. We can perform some very interesting experiments, and you can express some genes. Maybe you don't know where they come from who. I mean, who was the microbe containing that particular set of genes, but we can express this in molecular or in some cassettes and in other hosts, and we can have novel, for us, molecules. The last week, for instance, there was a paper in Cell showing a new antibiotic, discovered based on metagenomic data. So they actually didn't isolate the microbe, but they found a new set of genes in the environment, and they expressed this, and now they have a new antibiotic. So this is one approach, but this is really difficult and rare, and we have only a few % of success using this approach. And the most important thing, I believe, is still we still need to rely on bacterial and fungi isolation. So we need to isolate microbes. And nowadays, without that information, we can rely on very powerful approaches that we have, we call high throughput isolation techniques. And if you are curious about some of those approaches, I recommend to search this Schultz et al. From my group. We have actually 2 publications showing this, reviews in 2023, this year, different approaches that you can use, especially for extreme environments, and how can we extract or isolate more microbes from those very interesting environments. And what are the outcomes of all those powerful tools that we have nowadays? I mean, we can now describe novel species and novel strains. We can search for novel enzymes. So we have what we call extremozymes, and they usually have a very important application in several different companies or industrial applications. New drugs, I just mentioned this new antibiotic that was discovered last week. Improvement of agricultural practices. When we think about space research, so those microbes, in particular the microbes from extreme environments, they are amazing macrobiological model for, I mean, who knows? I mean, if there is life as we know it on Mars nowadays, let's say in the subsurface, because there on Mars you have radiation, a lot of radiation, the extremes of temperature. It's a very oligotrophic environment, the high level of CO2, but there are some ice on the subsurface and the protection from UV. So people from NASA or from the astrobiological program in China and some other colleagues, and I mean, I believe that there is a huge potential if there is microbial life on Mars. Nowadays, maybe they can survive on subsurface. But we don't have those microbes. There is a rover now trying to collect samples, but those samples will be bring back to Earth like in the next 10, 12, I don't know how many years. But we can try to understand the potential life forms that we can have on Mars subsurface or some other moons, like the moons on Jupiter or Saturn. Studying, those microbes that we can isolate from extreme environments on our planet in hydrothermal vents or desert or volcanoes, because they can survive in very extreme conditions that are kind of similar analogs to conditions that we can find in some of those places beyond Earth. And again, the huge biotech potential to improve industry process that we can have when we start to isolate and to study, especially like for the lecture today is about extreme environments and extremophiles. So in particular, the extreme extremophiles, the microbes that can thrive in very extreme environments. And one important thing to note is what is extreme? So I mean, we always have our anthropocentric point of view. So we consider extreme the condition that we cannot cope with. So we cannot survive in very extreme hot environment or in a very extremely cold environment. We cannot survive for a while. But there are organisms, especially microbes, that they can they love what we call extreme. And this is a table that I found very interesting. So those are the limits of life that as we know today. And I can tell you that every day or every week or every year, we have new publications, because I mentioned that we are using a lot of molecular tools and bioinformatics and, powerful tools that we can use nowadays, and we can extract a lot of information. So we are adding a lot of new information on this table. And so the limits of life, I can tell you for sure that we still don't know. But so far, the limits of life, for instance, in a high temperature environment, a thermophilic environment, some microbes that were isolated from hydrothermal vents. Hydrothermal vent, it's a volcano or, similar to a volcano. So we have a volcanic activity in the deep ocean, and you have this like a small volcano in the deep sea. And we can face very extreme hot temperatures around the wall of the vent, but we can have a gradient of temperature. And so there are some microbes, archaeas, including some bacteria, that can not only survive, but they can thrive, and they need high temperature to multiply, to divide their cells. And like boiling water, or microbes that can thrive on 121 degrees Celsius. So they can survive, they can divide the cells. And 121 for microbiologists, we know, is the temperature that we use in the autoclave to sterilize from normal mesophilic microbes. But if you have some extreme thermophilic microbe, hyperthermophilic microbe, like this one, it'd be nothing. It would be like, wow, that's amazing temperature. 121 is perfect. Also with the pressure, because those microbes, they thrive and they live in a very, remember, deep ocean. So it's also used to support a very huge pressure, atmospheric pressure. Low temperatures, I mean, there are some fungi, yeast actually, that was isolated from Antarctica environment in Antarctica. And the yeast can actually thrive and have metabolism at -20. So -20 degrees Celsius, the temperature that we use in the fridge to froze in some samples, those yeasts or even some bacteria, a lot of bacteria. Now we have a lot of information, some microbes, some bacteria that like actually this very extreme cold temperature. I mean, they will not go to sleep, you know, when they are at -20. They still perform metabolism. They are still active on such temperature. So, I mean, it's impressive. It's unbelievable. High concentration of salts, like in brine pools, we can have microbes surviving or even thriving. I mean, sometimes they need very high concentration of salt. So it's really a brine. It's really a, you know, extreme, the salinity is unbelievable. Radiation, there are some microbes. Deinococcus radiodurans is the most known microbe. It's the model for radiation tolerance. They can stand very strong doses of radiation that could kill a human in seconds. You know, it's a very extreme doses of radiation, but those microbes, they are happy with this extreme dose of radiation because they have particular mechanisms. They can rearrange, they can, you know, or reorganize their material, the genetic material, and they will be super happy with this huge dose of radiation. Those microbes are super interesting when you think about space exploration, but I will come back there. And actually, I must add that we have some microbes isolated from Saudi Arabia extreme environments that might be even more resistant than the model Deinococcus radiodurans. So it's just to showcase how important it is to explore the diversity of extreme environments in these thermophiles. We are always getting new information, adding new information on this framework. The best example of a thermophilic microbe that I think everybody knows is the Thermus aquaticus. It's a thermophilic bacteria. Was isolated in the end of 60s and beginning of 70s by Thomas Brock, famous, everybody knows the microbiologists, at least they know the Brock book and some other. It's a very, it was a very famous microbiologist, and isolated the Thermus aquaticus from the Yellowstone National Park in a hydrothermal vent in Yellowstone in US. And in that time, it was impressive to see a thermophilic microbe that could thrive in a very hot spring, in a hot place. And actually, we all know that most of labs nowadays rely and use a PCR, polymerase chain reaction, that actually was based on the DNA polymerase of this very interesting thermophilic microbe. And actually, the technique, the PCR, the polymerase chain reaction, gave the Nobel Prize to Millis in the 80s. So this is the most known application of a thermophilic or a thermophilic microbe. And of course, okay. I thought that was a question.
No, it's
okay, no, that's good. Yeah, and that's good actually, because my idea is to keep very informal the talk. I mean, it's not formal. It's like we can talk about these extremophiles. I love to talk about this subject, so it's really amazing. And the Thermus aquaticus, it was not the most impressive, I should say. It's super impressive, and the technology, the technique of PCR that changed, and it was actually a revolution in our biological sciences. Now we use PCR for forensic medicine and agriculture, and to study the soil diversity and human microbiome. So it's really a game changer, I should say. But nowadays, there are other applications of microbes. And going back to the Yellowstone, I just select, I'm not part of the company, so I'm not doing any marketing. I'm not getting any money from this. But I found this a very interesting example of new applications of thermophilic microbes. A fungi was also isolated from the Yellowstone hot spring, a very hot spring. And some scientists, they discovered that fungi, in the beginning, they were trying to use that fungi for biofuel production. It's a thermophilic fungi, and there was producing some, a potential biofuel, and so they were trying to perform this. But it was okay, but they realized that actually this particular fungi, thermophilic fungi, was really important in the production of biomass, microbial biomass. So that means a lot of potential food, vegan food, actually, totally vegan, because you have nothing, only the microbial biomass. And they came with this it's a Fusarium flavolaps, and the fungi, and they actually extract the biomass of this fungi, and they start to process this and trying to develop some different things like cookies, or the base for production of some bread. And also now they have this hamburger that the meat actually is made from Fusarium, some fungi. And so this is a very interesting approach, actually, when we think about space food for the astronauts, because they don't have, the possibility to have a huge plantation there, or for agriculture, or they don't have ways to raise cattle or pigs or chicken, food. And they can actually think about use of microbes as a food source. And microbes like this Fusarium species, they produce a lot of biomass. And the biomass can be, after some processing, they can be used for production of a very interesting food. And this is actually a huge company now. In less than, and this is why I brought this as an example. I was really impressed when I was reading about this company, because I not make marketing of the company, so I will not with the name. But in less than 5 to 6 years, they jumped from a few $1 million investment to $500 million investment in the company. So it's really impressive. The importance of one, thermophilic fungi and fungi, and the connection with biotech food production. And it's super interesting because you can actually produce a lot of biomass without relying on a huge amount of land, because we just need a huge factory, so we don't need farms to raise the fungi. And we can have this, the CO2, because the fungi is also using the CO2 fixing carbon to produce the biomass. So, so it's a win-win game. It's a very promising approach. There are a lot of new startups and some research on this particular field. And again, the microbes from extreme environments, they can play a very important role in this also food application. My experience with extreme microbes came from a long time ago. And actually, I was working still in Brazil with some heavily contaminated salt mangroves. So the mangroves in the northern coast of Brazil, and some of those mangroves, they can have a very high concentration of salt. Usually they are, of course, but you have a variation depending on the region. But some mangroves, especially in the northeast of Brazil, they have a high rate of evaporation and less water coming from rivers. And so they are quite saline, not as saline as the Red Sea mangroves here in Saudi Arabia. They are even more extreme. But extreme environments, so we have salinity and also high concentration of solvents, because it was a mangrove that was heavily contaminated with oil. It was the Brazilian oil company. They were trying to recover that mangrove, and they were trying to plant revegetation, planting mangrove trees in that mangrove. But it was so heavily contaminated that the mortality of the plants were like 95%. So it was almost impossible. And so we came with this biotech and bioremediation application. And we assembled a consortium of microbes. So we select some microbes, some bacteria that were able to degrade different fractions of oil. Plus, you know very well those microbes that can promote also the plant growth. So the plant growth promoting rhizobacteria. So we select some bacteria, some microbes from the rhizosphere of the mangrove plants, trees, and we combine those microbes with some other microbes that were able to degrade different fractions of oil. We encapsulated these to make some small microbial pills. So this is a polymer, a slow-release polymer, similar polymers that we can use for biofertilization in agricultural fields. So it was a very interesting approach because remember, in the mangrove, you have the tides problem. So the tide come and you have water, and this could wash our inoculum, microbial inoculum. So our solution was to encapsulate the consortia, into those polymers, slow-release polymers. And we tested in different ways. So in a mangrove greenhouse and inoculation on the mangroves, and we performed the experiments in the field. We applied the microbes, the consortia, and we planted again in the field, in the real field. And it was amazing. After 2.5 years, almost 3 years, finally, we start to, recover that mangrove using a combination of extreme microbes and plant growth promoting microbes. And this was super impressive because if you have an oil spill in a mangrove, there are some publications showing that if you leave the oil there, that mangrove can take 25 to 30 years to recover from the oil spill. Using our biomediation approach, oil degraders plus plant growth promoting microbes combined, we could recover in 10% of this time. So it was really impressive. We have a patent on this. We still have some papers going. And these also, of course, came with the seminal papers from my one student, Paketani. In 2009, we published some papers showing the potential, select the microbes. And after this, we went through the whole process. And so it was a very good example. Going to more extreme environment, I did a lot of work actually in Antarctica. Actually, the first huge survey, diversity survey of microbes from the terrestrial environment in Antarctica was for my group at that time. We published in 2010 a paper in ISME journal using for the first time in Antarctica sample a next generation sequencing. In that time actually was a 454 from Roche that nobody's using anymore, but in that time it was the best thing. It was the ultimate tool. And this is something that I'm really proud of. I was studying the microbial diversity from Antarctica for more than 15 years, 16 years. So I knew all those microbes from the terrestrial environment. And as you may know, in Antarctica, we don't have cities, we don't have people actually living there as we have in the continent, but we have the scientific stations. And so we have people living in scientific stations. And all those scientific stations, they try to not impact the environment, but there is always impact when you have humans living in any environment. So there is impact in Antarctica as well. And in 2012, there was a huge fire in the Brazilian Antarctic Station, and this was really bad. Some people passed away. I knew one of the guys. It was a pretty sad moment. And also the diesel, the oil diesel that is used, it was used to power the station, to heat the station, to keep the station working. It just spilled in the soil. So it was a real nightmare. So we had a huge diesel spill. The burn of the station, the 2 people passed away from this very sad moment. And there's no other way to clean the oil in Antarctica but the microbes. we had a lot of information on microbes and the diversity and the potential for oil degradation, because I was studying that time psychrophilic microbes, those microbes that like very cold environment. And I always been working with oil degradation since my time in Brazil. So I knew those microbes. So I said, well, let's put together a protocol to perform bioremediation in this particular environment. And it was a successful case based on our research. The Environmental Ministry of Brazil, they put together a very good protocol. And actually my postdoc in that time was leading the microbiological, one of the leaders of the microbiological work. So they performed the bioremediation there in the region, in the area, in the Brazilian station. And it was clean. So now we have a new station in that particular place. But again, using extreme microbes, so microbes that were adapted to cold situation, that actually we combined some microbes that could perform well in a very extreme, below zero temperatures. And they are still active, metabolically active, and so they can still perform the degradation of the oil. And it was a very successful case. We performed a lot of metagenomics in those samples. This is a shotgun metagenomics, just showing the potential function, so detecting the genes on the microbial populations from those soils. So we detect particular groups of microbes that were able to degrade. You know, the diesel oil, we have a lot of aromatic compounds. And so we selected those microbes that were able to degrade aromatic compounds, so that are very predominant in the diesel oil. And actually, we discovered that in that particular region of Antarctica, the degradation, the main degradation pathway for aromatic compounds is totally different from the metabolic pathway for aromatic degradation, biodegradation in any other environment. So it was quite interesting. So it's a very particular microbial community, you know, from that particular environment that was really able to degrade that oil. What we did in our lab was to help the environment. So we select the best microbes. We perform like enrichment of those microbes in the lab, and we put back on the field, the soil. So the degradation was a success. We also detect metabolic activity at minus 20 degrees Celsius. Again, confirming that we have a lot of microbes that can actually degrade oil, even during the winter time. There was a kind of, wow, in that time people said, oh, we can only perform bioremediation during the short window of summertime in Antarctica. In the window of short summertime, I'm talking about -5, 0, -1, you know, so it's still very cold. But in the winter time, the temperatures can go below 20, -30, -40. But we detect microbial activity at -20. So it's still degrading oil, those microbes. So, you know, this was a very important outcome from our paper, our studies, because we knew that we could perform engineering the bioremediation for a longer time. So we saved some time to clean up that soil. We isolate some microbes from another extreme environment in Antarctica, but this is also a paradox because it's a very hot environment. It's a volcano, active volcano in Antarctica. It's a deception island. We isolated several interesting microbes in producing biosurfactants, pigments, and we are still studying those microbes. We assemble a huge collection of very interesting extremophiles in my lab. So we have a collection here in my lab here in Saudi Arabia with hundreds, thousands of microbes from Antarctica, because I brought those microbes from to Saudi Arabia. So we have a lot of psychrophilic and thermophilic microbes from a volcanic island. And we have now thousands of novel isolates from different deserts, hot springs in Saudi Arabia, Pink Lake, there is a salt lake that we found here in Saudi Arabia, hydrothermal vents, now the first hydrothermal vent that is being described now in the Red Sea. We are helping the geologists studying the microbiome of those hydrothermal vents, deserts, and also I mentioned the hot springs and the volcanoes. We have more than 2,000 volcanoes in Saudi Arabia. Of course, most of them are inactive volcanoes, but some of them are still considered active volcanoes, but there is no eruption for a long time. But we have the heat, we have, all the power that we found in volcanoes, and we isolate some very interesting microbes on those volcanoes. And we are screening the collection now, and we are trying to search and to find some biotech applications, and also to describe novel genus, novel species of bacteria. And we have a lot of novel microbes here. And one example is a paper, a set of 3 papers. We isolated one microbacterium from Antarctica. We published the genome. We detect genes involved in carotenoid production in that particular strain. We extract, we characterize the carotenoid, it's a novel carotenoid, and we apply these in culture human cells to check the toxicity and the protection factor against UV radiation, because those pigments can protect those microbes from radiation in those extreme environments, UVC, UVB radiation. So we said, well, we thought, well, let's try to do this, and we extract this, and it was a very interesting results, like 50 times more protective than like a commercial sunscreen that has been used nowadays. So it was really powerful protection. And now we are trying to prepare some ways to deliver these pigment as like a novel sunscreen. So this is one particular application. We describe some novel species and actually a novel family of microbe, a thermophilic microbe, Carbonoactinosporaceae. And so we have a Carbonoactinospora thermototrophica. There is a new genus. I myself went to the lab and I was able to isolate one very interesting microbe from these very interesting consortia. So we published this year in Communications Biology, oligotrophic, extreme autotrophic consortia for microbes. And we detect carboxysome. So it's a consortia of microbes that is fixing carbon from the atmosphere. So it's only feeding on trace gas. So we don't add glucose, or any other, carbon source, only gas. So carbon monoxide, carbon dioxide, that's it. No nitrogen source at all. Also nitrogen, atmospheric nitrogen. So carbon monoxide, carbon dioxide, and nitrogen, and oxygen. That's it. In a minimal medium, it's almost water and some salts, and that particular microbe can thrive in that environment. It's only, it's fixing carbon, producing biomass, it's fixing or assimilating nitrogen. We are still trying to figure out the mechanism of assimilation of the nitrogen, and it's producing biomass. And this is a potential microbe that can really be applied like in production of biofuel, or maybe also in biofood. So it's extreme microbe with a huge potential. Here in Saudi Arabia, we have several volcanoes, several extreme environments, and desert, not only one, but rock desert, sandy desert, snow in Tabuk region in the north part of Saudi Arabia. So we can also have a cold desert here. Hot springs, there are more than 18, maybe 20 hot springs in the terrestrial environment. And now also here at KAUST, we are publishing, describing the first hydrothermal vents in the Red Sea. So, that's amazing landscape, very different extreme environments. So it's a real paradise to study thermophiles. So those pictures from the extreme environments, a hot spring and volcanoes, it's a beautiful, amazing place. There is one amazing, one of my favorite places in Saudi Arabia. It's Alula. It's in the north part of Saudi Arabia. It's a heritage site. It's very interesting. They have temple, similar carved in stones, similar to what we have in Petra in Jordan. So it's the same civilization. So this is Hega. So it's a very beautiful and important place here in Saudi Arabia. And we were able to sample the desert in that region. And we isolated several microbes. So we have more than 1,000 isolates from the Alula region. It's a very interesting desert spot. And as you can see in those pictures from Junior, we have a lot of pigmented microbes. Again, remember, in deserts, and usually you have extreme exposure to radiation. We don't have the cover that we have in a forest to protect against the UVB, or also desert. It's a very dry. You have the drought environment, so it's a very extreme environment, similar to what we can find in some other places like Mars, maybe. So a lot of pigmented microbes. We are studying those pigments now because those pigments, they have a potential use and application in industry, like a cosmetic industry. Some pigments actually are biosurfactants, and some of pigments are actually they have antibiotic properties, so they can be a novel antibiotic. So we are studying the chemical of those pigments now in my lab. And also we have a deep interest in the UV radiation resistance. So the radiation resistance. So we are developing some novel protocols to characterize those microbes. And this actually goes to the space research connection that I have in my lab. So we are trying to isolate very extreme microbes resistance to high doses of radiation. The pigments can be applied in several applications, as I mentioned, cosmetic and also pharmaceutical industry. So they are very interesting. And we isolated some very, we also working on, we don't have the Mars soil on Earth, but there are some companies that they develop a kind of simulant. So a regolith from Mars, they don't have organic matter there on the Mars, so it's a regolith. And we are testing the extreme microbes and how they can grow and how they maybe can improve the quality of those regoliths from that we can have on Mars, similar to what we have on Mars. And maybe in the future, this could be used for agriculture experiments and so on. And we can also maybe use the same microbes here on arid environments like we have in Saudi Arabia. So it's not only the space thing, but it's actually would be easier, you know, to apply those microbes here on Earth. Yeah, a lot of UV radiation, radiation-resistant microbes. We are performing some. We are also characterizing the metabolites of the microbes that are growing on those Mars simulants, the Mars regoliths. And I like this picture because the first one, Mars. The second one, Tabuk Desert region in Saudi Arabia. So it's amazing. I mean, they are very visually similar environments. Of course, there are a lot of difference. Tabuk can be very tough, but of course, Mars is even worse. But I like that picture a lot. We have a collaboration, a very strong collaboration with some NASA groups, trying to understand those microbes that we can have thriving on South extreme environments, because they can be very important models to try to understand the microbes that maybe we can find somewhere beyond Earth, or we can also use those microbes to test protocols to be sure that those samples, soil samples that they will bring back to Earth will not contain any potential harmful microbe, because we don't know. I mean, we have no idea. So it's better safe than sorry, as usual. So we are using also those extremophiles as model to treat decontamination protocols as well, in collaboration with NASA. In my lab, we also here in KAUST, we also trying to put together a Mars simulator chamber. So it's a small device actually that we can actually will be able to simulate Mars conditions. So this will be ultimate test for our microbes isolated from extreme environment in Saudi Arabia, if they can really cope with some very extreme conditions. And I just will pinpoint this particular case. It's a black fungi that was isolated from Al Haba crater. It's a huge, amazing, beautiful volcanic crater that we have here in Saudi Arabia. As you can see in that picture, in this picture, we have the crater, and there is a salt, high layer of salt, in the bottom of the crater. And it's a very extreme environment. Drought, salinity super high, and the radiation level, the exposure of radiation, it's it's there. There is no cover, no plant, nothing. But we isolated several very interesting bacteria, and this particular fungi, it's amazing. It's extremely resistant to radiation, more than the Deinococcus radiodurans. And we performed some tests adding that fungi in the Mars simulant soil, the Mars regolith. And we are studying the metabolites that fungi is producing in that particular very extreme environment, in that soil. And we also add a substance, a perchlorate. Perchlorate is highly toxic to all life forms here on Earth. It's highly toxic. We don't have, fortunately, in high concentrations here on our planet, but on Mars, there are a lot of perchlorate on the soil, on the surface. But we have here a fungi that is resistance to radiation and extremely resistant to high doses, high concentration of perchlorate. So this is, wow. So this is a kind of example that why NASA is so interested in our studies, in our collaboration. We also are studying using the supercomputer that we have here, Shaheen. We are studying the genomes of bacteria and fungi that were isolated from the International Space Station. So this is also a collaboration. So we are studying now more than 3,000 genomes from bacteria and fungi that were previously isolated from the International Space Station. And we are sequencing now more than 1,000 genomes. So more 1,000 genomes from the Phoenix mission to add on this database. And we are studying those microbes. And just one example, why it's so important to study those microbes that we isolate from the International Space Station. So this is a Klebsiella pneumoniae. It's a pathogenic bacteria. And actually, this one was a dominant strain in the International Space Station in that time. And studying its genome, we noticed that it's a hypervirulent strain. So we need to understand how the microgravity and how the radiation, because in the International Space Station, they don't have the atmosphere, that filter, because here we have the UVB, that is very important, and we need to use the sunscreen. But after the atmosphere, we have even worse, we have UVC and also some other cosmic rays. But the UVC, this is extremely mutagenic. We don't have it here on our surface, on our planet, but we have in the atmosphere. And the International Space Station is there. So they are submitted to high doses of radiation every day. And when you think about missions to deep space, now people are discussing how to send again astronauts to Moon and maybe Mars. So we are talking about deep space travel, so months. And to Mars will be years traveling into space. So we need to know more about the radiation effect and the microgravity effect on those in the DNA, and we can use the microbes to understand that. In this particular case, it was super interesting because imagine, an astronaut, they don't have a drugstore, so they cannot, use, or they don't have a hospital there. So it's a very tricky situation. So we need to understand more about those microbes. We can also study the genomes and also the metagenomic data from the International Space Station to extract information about potential industrial applications of those microbes. So this is something that we also study. thousands of genomes and metagenomes, and we are comparing these with thousands of metagenomes in the global database. And very meaningful information as well. Those are some other environments from Saudi Arabia, so it's not only desert or volcanoes, also the Red Sea is quite interesting. And I mentioned that we are studying the microbiome of the first hydrothermal vent. So those are two students, Nicholas and Sharifa. So they are studying, Sharifa is actually studying the microbiome of the hydrothermal vent. There's some very amazing data. We have a paper, a publication now under review that I think will be published at the end of this year. So the first look into the microbiome of the hydrothermal vents in the Red Sea. I'm also studying the microbes from the extreme mangroves that we have here in Saudi Arabia, so in this Al-Hajj region. And this is a very interesting, because the mangroves here are super extreme. There are no rivers, so the salinity is really higher than in any other mangrove. And so they have a unique microbes there. And we are studying those microbes, the metagenomic data, and actually, again, so we have the fundamental, the basic research, but we also try to apply that acknowledgement in the biotech applications. So we have a student, Hamad, he's actually, with help of other members of my group, trying to understand, select some probiotics, some good microbes that can help the growth of those plants, the mangrove plants in that particular extreme environment. So the mangroves will grow, but we believe that with some help of good microbes, they will grow faster and healthier, you know? And this is this will be online with the Saudi initiative to revegetation, to restore a huge amount of mangroves in the coast. So to finish my talk, we are now living in a very important moment from the microbiome field. I think we are really moving, to another frontier now. And the microbes and the microbiome, they are playing a very important role nowadays, because the microbes, they have this huge capillarity. They can purpose, they can go to different fields. So we can talk about microbes in humans and human health. We can talk about microbes, the importance into plant microbiome and the plant health, or in plant disease. How can we try to control using modulation of the microbiome, and how this can help in plant disease treatments, and also the corals and the ongoing research here at KAUST. My wife actually is working on coral microbiome, and she's using the good microbes, the coral microbiome, to protect the corals from bleaching. So, a lot of potential applications, and especially for the students. For an old guy like myself, I'm extremely happy to be working and to be living in this time, to see all those advancements, all those new tools. And if you are a young student, you should be super happy, because it's a very exciting time to get involved into microbiome research. It's really amazing. The tools that we have nowadays, we didn't add, 5 years ago, 6 years ago, 10 years ago. So we have a great potential. I truly believe that we have a great potential to improve human health, agricultural production, bioenergy generation, environmental sustainability. That is a very important issue nowadays. And even the microbes can help to conquer space. I'm pretty sure about that. And they are so important that I'm leading now a KAUST microbiome and microbiome science strategic plan that I hope will be will ignite, will be the catalytic point for Saudi microbiome initiative. That is super important, timely. We need to fill the gap that we have in this region. And I'd like to invite you all to join, at least virtually, a conference that I'm organizing this year, October 30 to November 1. This Trends in Microbiome in Digital One Health. this is a conference that we will have very important international guests, national guests as well. Some KAUST faculty also be presenting, and we will be touching all different aspects. So human microbiome, environmental microbiome, antimicrobial resistance genes, food microbiome, and of course the One Health and the digital One Health that is extremely important, especially for Saudi FDA. And you know, in every different centers nowadays. So you are invited to join this. I want to finish with this saying. It's a Bedouin proverb that I learned here in Saudi Arabia, and I think it's amazing. When you sleep in a house, your thoughts are as high as the ceiling. When you sleep outside, they are as high as the stars. So it's a very powerful saying from the Bedouins. And this, just to say that I do love to go in the field and to isolate those microbes. And I'm really blessed to be here. And this is my group, part at least of my group. We have more people, and it's always difficult to get everybody in the picture. So this is my email, my, I think is a Twitter, and the Twitter account of my lab, Magby Lab, and the website of my lab. There you can find more publications, some news, and always talking, always trying to spread the word about microbiome and science. With that, I leave you guys and open for if you have questions, something like that. And again, thank you very much for this amazing opportunity to talk about microbes. I do love this thing, this subject. Thank you. Yes.
And it is similar visual for us. So nice. So if
my sound is not so good.
Can
you hear me well?
You can.
No, I can't. It's you know, freezing the connection. Can you hear me well? I can't hear you.
Yeah, let it.
I can't.
Can you hear my voice right now?
Now I can hear you.
Oh, okay, Alex, can you hear my voice right now?
Yes, now I can. Now it's good.
Okay. Nice. Thank you. So, hello. Everybody, if you have a question, please raise your hands through, or you can put your question in the chat box. If you want, you can also talk in Indonesia. I will try to translate it for you. So it will make much easier for you to discuss further with Alex about microbiomes.
Yeah.
Especially for graduate students. The question is compulsory for you guys. Okay, Ali Budi Kusuma, please. You wanna propose by yourself? I cannot hear your voice, Ali.
Okay, can you hear my voice now?
Yes.
No, it's okay.
Okay. Hello, Professor Rosado. Thank you so much for your very nice talk.
Thank you.
So, okay, my name is Ali. I am from Indonesian Center for Extremophile Bioresources. And currently I am working with the UN, with the United Nations Environmental Program. So it's very interesting when, say, looking at your talk, especially when you discovered a novel yeast species, if I'm not mistaken, that they are resistant more than Deinococcus radiodurans, right?
Yeah.
And I would like to know more about genomic profile of this yeast. So whether or not you find a similar resistant genes toward radiations that you could find in Deinococcus, or they have completely different genomic profile. Like normally we have DNA repair mechanisms in such of strains. Do you find a similar mechanism that could explain your findings?
It's a very good question, and actually we are studying the genome of this particular yeast now, and what we know is that a new species as well. So it's a new species, and actually one of the mechanisms responsible for the protection to radiation, high, strongly and heavily doses of radiation, is the production of pigments, especially this melanin, a lot of melanin. So it's a black yeast. So this is helping, of course, a lot.
Sure.
And the genome, we were struggling in the beginning because it's a new genome. It took a while, and especially yeast. I mean, it's a little bit more complex than bacteria to understand.
Yeah.
But we are just now finishing. My student is analyzing this the genomes, and I hope to have more precise data in the next few weeks, I think, because you know, we found some very interesting portions of DNA, but the main factor for sure is the melanin production. So as you could see, I don't know if you remember in the picture, it's a very black yeast.
Black.
Yeah, and this is protecting a lot of against the radiation. We are still trying to figure out how this microbe can cope with high doses of perchlorate, because this it was unexpected. We have some clues now based on the metabolomics data, and we know that the fungi is producing some metabolites. Most of them are unknown, but so this is really tough to discover. But we are detecting some new tools, new features as well. Okay. I hope to give a better answer in the coming weeks or months.
That's really great. Yeah, that's really great. So this is the same strain that is resistant toward perchlorate exposure, right? Is it the same strain?
Yeah, it's very interesting strain. It was unexpected, and this was isolated from the crater, the volcanic crater.
You know, we did the same. Yeah, we isolated some actinomycetes from the caldera of the volcano here. And yeah, we're looking forward to establish a collaboration with your lab.
Oh, this would be wonderful. Yes, actinomycetes.
That'd be great.
Very impressive. Yeah, very impressive.
Yeah.
We have very cool. That particular microbe, extreme microbe that I was showing that can that is a new species and is thriving using trace gas. It's actinomycete.
Yeah. I've been following you on Twitter and Julia, and we are so really great with your work. Thank you so much for your today. Yeah.
Thank you very much.
Thank you.
Thank you.
Thank you so much, Ali, for your questions and also discussion. We need more collaboration with the microbiome expert from Saudi. And the next question, maybe from Valia.
Okay. Hello, Mr. Alexander. Thank you for the opportunity. My name is Valia Istifada. One of the slides explained that microbes from this extreme region can be used as a support in various industries. If the microbes that typically live in this extreme region are moved to the opposite or normal region, will these microbes still have the same potential? And will these microbes be weakened by changing their habitat? Thank you.
Yeah, welcome. Yeah, that's also a very good question. Yes, usually we can apply those microbes in particular industrial conditions. So when you go to industry, sometimes we have process that they need enzymes that will cope with very heat or hot process, hot conditions. And the extreme enzymes, they can actually support, and they can be performing super well in those particular extreme conditions that we can find in the industry. The same is true for the psychrophilic microbes. So we isolated some cold proteases that they can be very interesting when you think about some industrial process that they need to stabilize some products in in the refrigerator. And if you have some other enzymes or some process that will be performed in this cold situation, normal enzyme from a mesophilic microbe will have a very slow rate of processing. But when we apply a psychrophilic enzyme, that is enzyme that is used to work on a very cold temperature environments, that enzyme will perform amazingly well. So it's the same thing as we found in the Antarctica for the bioremediation. Remember the example I gave you. So we isolate microbes from the environment. So it's a very cold environment, and we apply it back in the cold environment. So it was performing the bioremediation. And nowadays we can also extract the enzyme. So we isolate the microbe, we extract the enzyme, and we can work with the extracts of the purified enzyme. So we can go to to apply this in different process. And the application of external microbes, extremophiles, it's really growing nowadays. And I can tell you, when we look into the biotech companies, the data on biotech companies, from the last 2 years to 3 years, they are booming in application of microbes on extreme environments. Because we had the Taq polymerase from the PCR in the beginning. After that, we had a kind of lack. There are some publications and applications, of course, but it's quite difficult. It was quite difficult to isolate and to study extreme environments. But with those new tools, we can really, isolate more microbes and understand better how they function in their extreme environments. And so we now can really boost the biotech applications of those extreme microbes. I don't know if I answered your question, but yeah, so thank you.
Thank you, Valia, for the question. Do you have another questions, or is it okay for you for now? Halo, Valia.
Iya, Pak, sudah cukup, Pak, sudah cukup menjawab.
Okay, thank you, Valia. Yeah. So the next question from another team. So Alex, actually, I also wondering about you just showed to us about the corona visualization, right?
Yeah.
For the metagenomic result, is it are they only the bacteria or fungus?
The
Community.
Which picture? I mean the.
I think you just showed us visual corona visualization graph that mention about biodiversity.
Okay. Yes, everything. Yeah.
Oh yes. Yeah, so I have a question for it. About the primers. What primer did you use so that we can get that detailed data? Did you design the primer for your own?
Yeah.
Or
For the amplicon sequencing, we rely on standardized protocols like from the Earth Microbiome project. So we follow the guidelines so we can really compare our data with their data. So it's a more global network. So it's better. For fungi, it's a bit tricky when you go for amplicon sequencing, because we have different possibilities, and I mean, yeah, it's in the database is also not that huge, and it's kind of tricky. It's difficult. But what we are doing now, it's mainly relying on metagenomic data. So we shotgun metagenomics. So we are not using primers at all. We are just sequencing, you know, the long reads. It's of course more expensive, you know, but you will have better quality data, because you will not only rely on taxonomical data, because you can say who is there, but with all those genes that we are sequencing without the bias of the PCR, of the primers, we can really detect the function of those microbes, and we can say, well, they are maybe involved in the nitrogen cycle or in the sulfur cycle using this and this metabolic pathway, you know, and this is what we are doing mainly in the lab now. And we are also trying to develop some new protocols for metabolomics in environmental samples, because it's also quite tricky, because metabolomics is a powerful approach, powerful tool, but it's still a long way to go. I mean, you know, we need to, especially when we are talking about environmental samples, because we have a lot of other things there, and how to be sure that we are detecting, you know, the metabolite from this and that, and the microbe and the biotic factors as well. So it's quite complex, but this is what we know. Yeah.
So I agree with you, because I try to analyze fungal diversity using general primer, and we used nanopore sequencer, and we cannot read about 30% of the sequence.
Yeah,
we are not able to see that. Maybe the shotgun approach is much better, as you say.
Yeah,
without PCR, but it's
exactly.
The price is higher.
Yeah, that's true. Exactly, that's the problem. I mean, so usually sometimes we rely on amplicon sequencing, so we can kind of filter our samples, and so we select the best ones, and we go for metagenomic sequencing. I mean, when we have a lot of samples, so we need to select, because it's literally almost impossible to you know, thousands of shotgun metagenomic sequencing. But we can kind of screen and select the most interesting samples, and there we try to do the metagenomics.
All right. So we have also a question from Samsul Huda and Buku. Do microbes isolated from extreme environment have special characteristics that are superior and dominant compared to microbes isolated from non-extreme areas? And is there a possibility that microbes isolated from extreme areas will not be able to collaborate with microbes isolated from non-extreme areas if they are united in the form of a consortium?
Yeah, it's a very interesting question, because we can mix microbiological, you know, fundamentals from microbiology. And that's true. I mean, usually depends. For some process, the extreme microbes, they are better, and they will perform, you know, better than mesophilic microbes, or especially those industrial applications that I mentioned, or you know, if you need to work in a biofuel production, sometimes you have processes or steps that you need some thermal application, and the enzymes or the microbes from a mesophilic microbe, they will not survive in those processes. So we really need the extreme microbes, and this they can perform better in very extreme conditions that we found everywhere in the industrial applications. So they are better than others. And the application of stromatophiles in microbial consortia, it's quite interesting, because this is also a kind of new, you know, field. We are, for instance, we are trying to develop some stromophilic consortia to degrade plastic in mangroves here, you know, so microplastic degradation. So we are trying to select some particular microbes that are able to degrade plastic under the high salinity concentration that we have here, especially in the northern mangroves in the Red Sea. And so far, some seems to be very potential for powerful approach. But yeah, so that depends. I mean, it depends on the case and the application. Sometimes they can perform better. Sometimes you don't need extreme microbes in your system, of course. Most of the cases you don't need. I think it depends pretty much on your goals, your ideas, or your target. And so, but when you need, they will perform better than any other microbe for sure. They are super tough microbes. I like to say that they are kind of superheroes in the microbiology.
Thank you. Maybe this will be the last questions as we have a limited time, guys. So from Salwa and Farid, how can Metagenomics help us understand the adaptation of extremophile microbes to the extreme condition in their environment? And how can this affect the development of science, especially in agriculture? Maybe if you want to give us some comments for it.
Yeah, I comment. I read some. Yeah, the metaomics. Yeah, the multiomics. I mean, we really need the multiomics, and sometimes can be really difficult to work on metagenomics or metatranscriptomics or metabolomics. I mean, especially when we talk and we go to extreme environments, because usually we have a low biomass environment, and this actually. I think it's the main challenge that we have when we go to extreme environment. So this is different when we, I mean, when we think about soil from a plantation, agricultural field. So usually you have a huge abundance of microbes. So it's easier to study those microbes. When you go to an extreme environment like a desert, hydrothermal vent or hot spring, usually you have a low biomass microbes. You have a particular group of microbes, microbial community, they are powerful, but they are not as abundant as microbes that we have in other more, with a lot of resource, like a forest or agriculture or soil or so on. And this, I think, is the main challenge, because you can have a lot of bias and you can waste a lot of time actually trying to extract the DNA and the metabolites and everything. But there are some very good tools nowadays and protocols that we can really obtain those microbes or the DNA or the metabolites from the environment. And if you are interested in extreme environments and those protocols, I recommend you to try to look into the name is XMP, Extreme Microbiome Project. That is led by Scott Teig. I'm also part of this group now. Scott Teig from Vermont University in the US, but it's actually a global consortium. So there are experts from different extreme environments, and we debate the best protocols, the best information that we can extract from different ecosystems and extreme environments. And so they have very good protocols. Sometimes we need to use a special cocktail of enzymes. Sometimes we need to rely on special protocol for DNA extraction. So now we are moving the field, I think, now better than a few years ago, and we can extract more information. So it's really the multiomics are going to a next step now based on those new protocols. Yeah, a lot of things to do yet, a lot of research, but we are moving now to really extract information and also to have products as well for biotech applications.
Yeah, thank you. Thank you so much, Alex. So everyone, I think just we have a limited time. Maybe I will try to conclude our lecture today about the understanding of how microbiome is critical, because using this microbiome concept, we can see much deeper about who are microbes that exist in one ecosystem, especially in an extreme location. As Alex said, that they are some of them can become a superhero yeah, they are anyway, they are living in the extreme situation. So we hope that they can solve extremes problems that maybe we need to face in the space or maybe some area. So if you want to also conclude something, Alex, please, maybe some conclusion from your lecture.
Yeah, no, just yeah.
Do you have one?
Yes, thank you.
Yeah. All right. So I think we're going to meet again next week on Monday, everybody. So don't miss this lecture with Alex to discuss further about microbiome. Alex will share the materials, the topics, so we can learn first and we can make more fruitful discussion in the next meeting. So thank you so much, Alex, and back to Miss Dian for the closing.
Thank you again. Thank you very much. It was amazing to see you guys again. It's my pleasure, always.
Yeah. Dian, monggo.
Yeah. Okay, thank you so much, Asibli, for leading a really good and also amazing and fruitful discussions with Alex. And then before we end the sessions today, I would like to remind all of you that we will have another sessions with Alex in the next 2 meetings. So please looking forward for that. And then once again, thank you so much for all the participation of the participants or the lectures and also the good presentation of our guest lecturer today, Alex. Thank you so much for that. And then I am from the international office. I would like to say that we are also welcome and open for another collaboration with you. And we are looking forward for that. I think that's all. Thank you so much for coming, everyone. See you again next time. Stay happy, stay healthy, and see you next week. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and good morning, Pak Alex. Thank you so much.
Thank you very much. And have a good day, guys. Thank you
so much.
Have a
good day. Thank you.
Bye-bye.
Thank you so much. See you next week. Bye-bye.
Thank you. Bye-bye.
Mungkin untuk adik-adik yang belum isi absen bisa absen sehingga bisa didapatkan untuk anunya ya sertifikatnya supaya tidak missing sertifikat. Baik adik-adik, terima kasih atas perhatiannya. Terima kasih Bu Dian, Bapak Ibu yang sudah hadir pada acara pada pagi hari ini. Saya tutup. Assalamualaikum warahmatullahi wabarakatuh.